So your exam is on Monday, Tuesday. You get back. I just wrote it. I think it's good. This might be a case for the multiple choice or potentially a little bit harder. Their free responses can only ask you so many things. So the free responses tend to be a little bit more general because of the way the topics fall, whereas the multiple choice can be specific. So I don't know. I just think you guys might prefer the free responses on this one, surprisingly enough. Topic-wise. Oh, say that word. Topic-wise. Enzymes, photosynthesis, and cellular respiration. For photosynthesis and cellular respiration, make sure you have your diagrams. I don't know if it's any of you that still have diagrams back there or not. And you should be able to fully explain your diagrams to someone else. Okay? Use your Thanksgiving guests as hostages while they're at the table and explain the process to them. My thought process was when the mashed potatoes come out, you can start with, well, how did the glucose get into the mashed potatoes? And just go from there while everybody has to stay seated and listen to you. You also need that one pager of notes, the C4 and CAM plants, and then the fermentation and anaerobic respiration, and then all of enzymes, which wasn't really that bad because we had learned a lot of it when we did proteins. Okay, so that's topic-wise. All right, so our first free response that we're just going to practice is on enzymes. It got cut off a little bit if you're following along on your screen. We started a little diagram. Sucrose can become glucose, and glucose can become sucrose. We've got two arrows. Glucose can become starch. All right, that's not terribly surprising to you, hopefully going into this. We see we've got two enzymes, sucrose synthase and starch synthase. Before I've read anything, how do I know that those are both enzymes? Enzynase. Okay, so I know I've got an enzyme to turn sucrose into glucose and then glucose into starch. Okay, a rice grain is a fruit. There's your fun fact of the day. You could also bring that up at Thanksgiving dinner. It's not a fruit like in terms of the nutritional value, but it is a fruit because it has a seed in it. Most of the dry mass of rice grain is starch. In rice plants, starch is produced by hydrolyzing. What's hydrolyzing mean? Breaking with water, better known as hydrolysis, but hydrolyzing, you'll use it in a sentence. By hydrolyzing sucrose and then linking the released glucose molecules together into starch. They don't say it, but what would that be called? Linking the glucose molecules. Dehydration synthesis, very good. In figure 1, you see that. The optimal temperature range for starch synthase activity in a particular strain of rice is 27 degrees to 30 degrees. The optimal temperature range for the sucrose synthase is 30 to 35. Now, you guys can always approach these questions however you want. When I do this one myself, I do actually write the numbers up under the enzyme because it feels like they're trying to trick me by the fact that they keep talking about them backwards. They talk about the second enzyme first and then the first enzyme second. So do what you want, but I always write the temperatures right underneath. Okay, then the actual question. Describe how temperatures above 35 degrees C most likely affect the structure and function of the starch synthase in a particular strain. Using the information provided, predict the most likely consequence to starch content in mature rice grains if the rice is grown in an area where average temperature is 33 C. So, take 2 or 3 minutes, turn to somebody near you, and just verbally discuss how would you answer this question. Go.
Yeah, because it goes from very, very high. Really? Because this is temperature is 30, 35, and it's asking how what happened. Yeah, because the sucrose. Yeah, it's too hot. Yeah, too much starch, whatever.
Cute though. Very cute. Yeah, I mean, yeah. I'll share.
That's what I think.
Yeah. Oh, cool. That's cool.
Okay, you've all stopped talking. Not that you necessarily really started in the first place, but you've stopped mumbling. So let's see what we're willing to come up with. All right, describe how temperature is above. So before we tackle this, is this a long or short free response question?
Short.
It's a short. Short are always worth how many points? 3 or 4. Before you go in, it is a good idea to know how many points you think you're trying to earn. What's your best guess? Is this a 3-pointer or a 4? Is it 3 points? How are they allotted? We've got 2 words in bold. Describe and predict. Which one's worth 2? Describe is worth 2. 1 for structure, 1 for function, and then 1 for prediction. All right, so let's start with structure. What is going to happen to the structure of the starch synthase at 35 degrees? Do you not want your voice on camera, or do you just not want to participate at all? Liam.
It'll be denatured.
It'll be denatured. Okay, denatured is a good keyword. You're going to need to be in there to be full credit, but you would not get credit if you just say it's denatured. What have you not done? You haven't, well, we're still on structure. Why is denatured not enough for structure? Due to denaturing, the active site would change. That's good. That's better. Yep.
Did you all say like tertiary and quaternary structure?
That's the words you're going to want in there. Tertiary, well, tertiary is yes. Quaternary is a maybe. So you don't have to include it, but there's one more you do have to include. Secondary. So you're going to want to say that the protein's going to denature, the enzyme's going to denature at that temperature, which is going to cause the secondary and tertiary, maybe quaternary, if you want to include it, structure to what? What word could we put in there? Alter, change, unfold, all of that is fine. Why? You don't necessarily have to put this level of detail, but why are those levels of structure changing? Let's make sure we remember. Or why do those levels break down and primary doesn't? It's primarily composed of hydrogen bonds. Be careful you don't say exclusively. There's also ionic bonds in there. There could be disulfide bridges, but it's primarily hydrogen bonds, and they are weak individually, and the temperature will break them down. Okay, since we're on it, why does higher temperature break hydrogen bonds? Say it again. Okay, and that more energy is going to break the bonds how? You're correct, but why? The more energy is going to be seen how? If we could see the atoms, we'd know they'd have more energy. Why? They're moving faster, and they're going to hit the point where they're locked into a structure. So they're going to start vibrating and vibrating faster and faster and faster until basically what happens? They break free of the bonds. The energy trying to move them becomes greater than the bond holding them together. They break free. We lose our structure. Does cold denature an enzyme? It stops it, but it does not denature. Good. Cold doesn't do anything to the tertiary structure, to the secondary structure. Structurally, the enzymes are just fine in the cold. But you're correct, the enzyme still doesn't really work, but it's obviously not denaturing. So why does the enzyme not work when it's cold? How does a substrate find the enzyme? Right? So they have to what? They have to what? Until they collide. They have to randomly collide. What happens when temperature goes down? They're moving way less or not at all if they're frozen. So they're not going to collide, or it's going to be much, much, much less likely they're going to collide. You guys know this internally from food. It's why we refrigerate food. It's why we freeze food. It maintains it in its current structure, right? When you put chicken into the fridge, as long as it's not sitting there for like months, it goes in and comes out looking very much the same, no structural changes. But when you cook it, it looks very, very different because cooking it denatured everything, changed the structure of the proteins. Refrigerating it didn't. Refrigerating it just prevents bacteria from doing anything to it because it's too cold for any of the enzymatic interactions. Okay, you've got 1 point. How do we get the second one? Let's talk function. So we know it denatured and we lost secondary and tertiary levels of structure, which could impact the shape of the active site. How does that impact function? How would you get the point there? Good. The change in shape indicates that the substrate either won't fit at all. You can put that as an option, meaning we're going to lose all function. Or you could say what? The substrate, you could say it doesn't fit at all or doesn't fit as well. So you don't always lose 100% of the function. Sometimes you just lose some. It doesn't fit as well and it's not going to be as effective. Or you could say it loses all structure, therefore it doesn't fit at all, and you lose all function. Both are correct answers. Just follow them all the way through to saying what's happening. Okay, so then predict the most likely consequence to starch content in mature rice grains if they're grown in an average temperature of 33 degrees C. Does that work for sucrose synthase? Yep, that's totally fine. Does that work for starch synthase? No. So how would you write this up? What would be your prediction for starch content in mature rice? Did you say reduce? I can't hear. Okay, that's good. I just couldn't hear you. Okay, starch content would be reduced because? Good. So be careful with your wording. So starch content could be reduced, and you said because the enzyme's denatured, it can no longer synthesize starch. Then it wouldn't be reduced, it would be gone. Just make a match. You can say reduced, and you could say because the enzyme doesn't work as effectively because it's been denatured, or it's gone because it doesn't work at all. Just make sure your two terms match. Good. And that would be a 3 out of 3. Questions? How do you feel about it? Okay. All right, do you guys want to start to look at the review sheet? Okay, pull it up. I can't put it on the screen, but if you guys would go ahead and pull up the review sheet, we can talk about it. Let me print one real quick so I can put it under the document camera. Got an error when I went to print it. That's always a good sign. Oh, yeah, I'm sorry. That's great if you have it. Thank you for sharing. I won't write on it. Give you secret notes? Probably that's the first couple, to be honest. Okay. Thank you. Okay. Unit 3, cellular energetics. I want to start by discussing this fitness one because everybody sees it, including teachers, and goes, huh? Why is that in this unit? It's evolution. You guys know survival of the fittest. So fitness is a measure of how able an organism is to live and thrive in a specific specific environment. The fitter you are, the more likely you are to survive for a long period of time in that environment. The less fit, the less likely, the more likely something else is going to eat you or compete with you. Okay? So what that's referring to in this unit is the CAM and C4 plants we talked about last class. So basically, those types of plants have evolved to be able to be in an environment that plants shouldn't be able to be in. And that's all it's talking about. Just at every level of biology, we can tie evolution in as to why are we seeing these types of organisms. Okay? All right. Enzyme structure, that's understanding that they are mostly made out of proteins, and then, primary, secondary, tertiary, quaternary levels of structure and how that works. Enzyme catalysis, explain how enzymes affect the rate of biological reactions. They make the rate faster or slower? Faster by lowering activation energy. Environmental impacts, explain how changes to the structure affect its function. We just did that. And how the cellular environment affects that activity. We did hot and cold. Let's do pH. What does pH stand for? Power of hydrogen. So how could changing the number of hydrogen ions, either to more or less in an environment, denature a protein? Why does the number of hydrogen ions make any difference to whether or not a protein enzyme is denatured? What do you think? Thompson?
That might, they might bond with other hydrogens.
Good. So hydrogen bonds are between a partially positive hydrogen and a partially negative something. If we start messing with the number of hydrogen ions in the environment, you now have a lot more or a lot less positive things floating around that'll not just impact your hydrogen bonds, but also impact your ionic bonds. It either is going to start to pull things apart. That tends to be when you increase the number of hydrogen ions, which is actually decreasing the pH. Remember, there are opposites. Okay? And when you decrease the number of hydrogen ions, you actually usually get the protein shriveling up on itself because the bonds actually become stronger because nothing's pulling on them from the outside. Okay? The other environmental impact is salt content. This is not salt like table salt. This is salt like chemistry, meaning anything that's ionically bonded. It's the exact same thing then as hydrogen ion content or pH, because it basically means you're messing with the number of ions in the environment, both positive and negative. You put more or less than are typically there, you're going to get things shifting around in the three-dimensional structure. Okay? Cellular energy. That's the graphs we made. The free energy graphs. Can you identify the parts of and or can you create a free energy graph that shows an endergonic reaction versus an exergonic reaction? Can you show me where reactants versus products would be? Could you show me what it would look like if we added an enzyme and the activation energy was less? That's what that section is about. Can you? Okay. You can ask questions, so I mean as we go, if you don't understand any part of this. Photosynthesis. That's our drawing. Can you take your drawing and could you walk someone else through it without additional notes? Okay, and then can you discuss CAM and C4 plants? Cellular respiration, can you take that drawing, walk somebody through it without any additional notes? And then our outliers of anaerobic respiration and fermentation. And then fitness we talked about. Okay? All right. Let's look at some of the specific questions. What's the monomer that makes up an enzyme most of the time? Oh, come on. Amino acids. You should have all called that out. That was lame. Amino acids. How does the substrate bind to the enzyme? How does it find it? What did we say? It collides on purpose? Random collisions. That's why if you increase the amount of enzyme or increase the amount of substrate, it'll be more efficient because it's more likely they collide into each other. Okay? Or if you increase temperature to a point, it's more likely they collide into each other. What happens after the substrate binds to the enzyme? Any idea? Does the enzyme change shape when the substrate binds to it? A little bit. Does anybody know what it's called? Starts with an I. Induced fit. Basically, the substrate is bonding to the enzyme, right? So we know anytime you add or remove something from a protein, the shape does change. Doesn't have to change a lot, but it will change. So when the substrate comes into the active site of the enzyme, it effectively kind of hugs around it, locking it into place, and it's called induced fit. So it just does an awkward hug, okay? And then it's going to help it do whatever, either build or break bonds. Remember, enzymes can have multiple active sites. If the idea is to build, it could have an active site for like 2 or 3 reactants, and it helps put them together. If the idea is to break, it'll only have like 1 active site, and then the idea will be to break it apart. What's the function of an enzyme? It's not to speed up. It's technically to lower activation energy. Okay, so make sure there's a great byproduct of speeding things up, but that's what it technically does. Okay, true or false? Enzymes affect the Gibbs free energy of a chemical reaction. This is a good question. Say loud. No, false. Reactants, products, their energy levels never change. Okay, if enzymes affected free energy, we'd be breaking laws of thermodynamics, right? They make it go faster, they lower the activation energy, but the initial energy of the reactants, the final energy of the products, always the same. Can I have one more page? We'll do like 2 minutes and then we're done. Yeah, absolutely. Correct. Correct.
And then when you say how does it complete its function?
So that's tough because there's no simple answer to that, because it's either building or breaking bonds. That's probably the simplest answer you could give, but it's not going to be the same. Like if it's breaking bonds, they actually do put some physical tension on the bonds to help them be in the correct arrangement to break apart. If it's building, they hold everything in the correct orientation to make it more likely that they go together. Like that's why it's a tough thing to answer. Good. That was a good question, though. Okay, we already hit on this first one. How does an enzyme affect the rate of biological reactions? Makes them go faster. What is an enzyme catalyzed reaction? I mean, you guys understand that. That's just any reaction that uses an enzyme to lower activation energy. There are a very small handful of biological reactions that don't use enzymes, but honestly, most of them do. Even when we don't show them, most of them do use enzymes. How is the activation energy of an enzyme catalyzed an uncatalyzed reaction? We know the activation energy is going to be much higher in an uncatalyzed reaction. It's going to be lower in one that has an enzyme. How is the change in free energy of an enzyme catalyzed and uncatalyzed reaction different? It's not. That is a trick question. The change in free energy is the same. It's just a difference in activation energy. How is the reaction rate? We know an enzyme catalyzed reaction will be faster because it's easier to mount that activation energy amount versus an uncatalyzed will be slower. All right. Anything on what we've talked about today? All right, I believe we're done at 9:05, so we'll end that for today. All right.